Okay, let's unpack this. Today's deep dive is basically a conceptual crash course in the workhorses of the human body, enzymes. This foundational knowledge is, well, it's non-negotiable for first-year medical students. So we're really trying to distill the absolute highest yield stuff on structure, kinetics, and importantly, clinical diagnostics.
Yeah, exactly. We're aiming for real conceptual mastery here. Because honestly, understanding the functional machinery of the cell, it starts right here with enzymes. These are the biocatalysts synthesized by living cells. And they are overwhelmingly protein in nature.
Right, but we should remember that one key exception, the RNA-based ones.
Ribozymes.
Ribozymes, exactly. Good point. But mostly protein, and they are absolutely essential.
Their core job seems simple on the surface. Speed up a chemical reaction without being, permanently altered or consume themselves. I liked the analogy in our source material. Think of a really complex textbook that students might struggle with for days.
The teacher, that's the enzyme, simplifies the tough concept, speeds up the student's understanding, but the teacher is still there, ready for the next class, unchanged.
And that acceleration is just breathtaking, really. Think about protein hydrolysis. If you try to do that chemically, with strong acid, boiling heat, it takes days.
Right.
But the enzymes in your gut,
Yeah.
working at a gentle 37 degrees Celsius, they do the exact same job in just a few hours.
Wow.
Life, as we know it, simply doesn't function at a useful speed without these incredibly efficient biological machines.
Okay, let's understand this step by step then. Before we jump into what they do, we need to know what they are. So when an enzyme is fully functional, active, we call it a holoenzyme.
That's the one. The holoenzyme is the complete active package. It's got two main parts. First, there's the apoenzyme. That's the protein component, the part that gives the enzyme its structure and, crucially, its specificity.
Okay, the protein part.
But the apoenzyme alone usually isn't active. It needs a non-protein partner. If that partner is a small organic molecule, we call it a coenzyme.
Got it. Apoenzyme plus coenzyme. And just to clarify that structural language, sometimes that non-protein partner is really stuck on there, like tightly bound.
Exactly. If it's tightly, sometimes even covalently bound to the apoenzyme, we give it a specific name. Prosthetic group. Understanding this combo apoenzyme plus coenzyme or prosthetic group is key because the apoenzyme by itself is typically inactive.
And it wasn't obvious they were proteins initially, was it?
No, not at all. It actually wasn't until James Sumner crystallized urease back in 1926 that we definitively proved their protein nature, a major breakthrough.
So with thousands of different enzymes floating around doing different jobs, how did scientists even begin to organize this? It sounds like chaos.
Well, yeah, it could have been. But the International Union of Biochemistry, the IUB, stepped in. They created a standardized classification system based purely on the type of reaction the enzyme catalyzes. They settled on 6 major classes.
Okay, 6 classes. Can you run us through them? And maybe there's a trick to remembering them.
There absolutely is. The mnemonic everyone uses is OTHLIL. OTHLIL.
OHLL.
So the O is for oxidoreductases. They handle all oxidation reduction reactions, basically, electron transfers.
Makes sense.
Transferases. These enzymes move functional groups like a phosphate group or an amido group from one molecule to another. Hexokinase, which phosphorylates glucose, is a classic example.
Got it. H.
Hydrolysis. They break chemical bonds by adding water hydrolysis. Think lipases breaking down fats or pepsin breaking down proteins in the stomach.
Okay. L, the first L.
That's lyases. These enzymes add groups to double bond or remove groups to form double bonds, or sometimes they cleave CN bonds without hydrolysis or oxidation.
Right. Then they buy.
Isomerases. They just rearrange atoms within a molecule, interconverting isomers. Same chemical formula, different structure.
And the final L.
Ligases. Think to ligate or join. These are the builders. They join 2 molecules together, and this process usually requires energy, typically from ATP hydrolysis.
OTHLIL. Oxidoreductases, transferases, hydrolases, lyases, isomerases, ligases. Okay, locked in. Now, here's where it gets really interesting, I think. The action itself. How does this, massive protein actually find and grab onto its specific target, the substrate? We need to talk about the active site.
The active site, yes. It's really the functional heart of the enzyme. And it's not a huge part of the protein, usually just a small pocket or cleft. It's formed by specific amino acids, the catalytic residues. And what's amazing is how protein folding works. These critical residues might be really far apart in the linear amino acid sequence, like lysozyme, with residues like 35, 52, 62, 63, 101 involved. But the 3D folding, the tertiary structure, brings them precisely together to form that functional pocket.
So clever. Now, we often talk about two main ideas or models for how this substrate interaction happens. We started with the older one, the rigid lock and key model from Fisher.
Yeah, the lock and key model, it was a good starting point, suggesting, a pre-shaped rigid template in the enzyme that perfectly fits the substrate, like a key in a lock.
But that's not the full picture, is it?
No, it's a bit too simplistic. The reality, which Koshland described with his induced fit theory, is much more dynamic. The active site isn't rigid, it's flexible. The act of the substrate binding actually induces a subtle conformational change in the enzyme itself.
Oh, okay.
The tweaking achieves an even tighter fit and lines up the catalytic residues perfectly for the reaction. It's less like a rigid lock and more like a glove adjusting to a hand.
That makes more sense. Now, thinking about the energy involved, enzymes don't actually change the final balance, the equilibrium of a reaction. So how do they speed it up so dramatically?
They work by lowering the activation energy. That's the key. Imagine you need to get from one valley to another, but there's a huge mountain in between. That mountain peak represents the activation energy barrier.
Okay.
An uncatalyzed reaction has to climb all the way over that mountain. What the enzyme does is essentially dig a tunnel through the mountain.
A shortcut.
Exactly. The starting point and the ending point, the overall energy change of the reaction, are exactly the same. But the tunnel provides a much lower energy path, allowing the reaction to happen much, much faster, especially at body temperature, below 40 degrees C.
Brilliant analogy. Okay, that brings us neatly into kinetics and probably the most famous term here, the Michaelis-Menten constant, Karamiram. This value is just fundamental for predicting how an enzyme will behave. Let's nail down that definition again, and especially what its value tells us clinically.
Right. The Karamiram is formally defined as the substrate concentration S, at which the reaction velocity is exactly half of the maximum possible velocity, or Vmax2. Now pause for a moment and think about this. The real significance of Karamiram is what it tells you about the enzyme's affinity for its substrate.
Right.
It's an inverse relationship. A low Karamiram is a high affinity.
So a low Karamiram means the enzyme grabs the substrate really well.
Precisely. It means the enzyme is very efficient. It can find and bind its substrate effectively even when the substrate concentration in the cell is quite low. That's a crucial concept.
Definitely. And to actually measure Vmax and Kalman accurately in the lab, we almost always use the Lineweaver-Burk plot. Why do we bother with that double reciprocal plot? The standard Michaelis-Menten curve seems simpler to look at.
Well, the problem with the standard hyperbolic curve plotting velocity versus substrate concentration is that it approaches Vmax asymptotically.
Meaning it gets closer and closer, but never quite touches that maximum speed line.
Exactly, which makes it really hard to pinpoint the exact value of Vmax and therefore Klam just by looking at the curve. The Lineweaver-Burk plot solves this. By plotting the reciprocals 1 over velocity versus 1 over substrate concentration, you get a straight line.
And straight lines are much easier to work with.
Much easier. You can extrapolate that line back to the axis. The y-intercept gives you 1 Vmax, and the x-intercept gives you a metric 1 kilometer. Simple algebra then gives you the precise values.
Okay, moving on. Like any chemical reaction, enzyme activity is sensitive to its environment. Let's talk about the main factors. Temperature and pH.
Sure. For temperature, most human enzymes show that classic bell-shaped activity curve. Activity increases with temperature up to a point.
The optimum.
Right, the optimum temperature, which for most human enzymes is around 35 to 40 degrees Celsius body temperature, essentially. But if you go much higher, say above 50 or 60 degrees, the heat energy starts to break the weak bonds holding the protein's 3D structure together.
Denaturation.
Denaturation, exactly. The enzyme unfolds, loses its active site shape, and activity is permanently lost.
And we see this principle applied medically, right?
Absolutely. Induced hypothermia is a great example. Cooling a patient's body down during, say, complex heart surgery deliberately slows down enzymatic activity and the overall metabolic rate. This reduces oxygen demand and buys the surgical team critical time.
Makes sense. And even just keeping food in the fridge.
Same principle. You're slowing down the enzymes and spoilage bacteria, preserving the food longer.
Okay, what about pH? Another bell curve.
Generally, yes, another bell-shaped curve, but the optimum pH varies enormously depending on the enzyme and where it works in the body. Pepsin, the stomach enzyme, works best in highly acidic conditions, pH 1 to 2. But alkaline phosphatase, found in bone and liver, prefers a very alkaline environment, pH 10 to 11. Huge difference.
Wow. Okay, so we talked earlier about the apoenzyme needing its partner, the coenzyme, to function. Let's circle back to those essential non-protein helpers. What's the critical link we need to remember between coenzymes and nutrition?
The absolute crucial link is the B-complex vitamins. Most coenzymes are derived directly from these water-soluble vitamins that you get from your diet.
Oh, okay. Like which ones?
More classic examples, NAD plus and NADP plus R, which are vital electron carriers, come from niacin, that's vitamin B3. FAD, another electron carrier, comes from riboflavin, vitamin B2. Their biochemical function in the body is their role as coenzymes, helping enzymes carry out reactions, often by transferring specific chemical groups like electrons or acyl groups.
That connection is really important clinically. Okay, let's shift gears to inhibition. This is huge in pharmacology, understanding how drugs work. We can start with reversible inhibition, specifically competitive inhibition.
Right. Competitive inhibitors usually look a lot like the enzyme's normal substrate, their structural
Yeah.
analogs. Because they resemble the substrate, they can actually bind to the enzyme's active site.
So they compete for the same spot.
Exactly. They directly compete with a natural substrate for binding. If the inhibitor gets there first, the substrate is blocked.
And kinetically, what does this do? What's the hallmark effect on chemium and Vmax?
With competitive inhibition, the apparent chemium increases. You need more substrate to reach half the maximum velocity because the substrate has to outcompete the inhibitor for the active site.
Okay, Km goes up. What about Vmax?
Vmax remains unchanged. If you flood the system with enough substrate, you can eventually overcome the inhibitor completely and reach the same maximum speed.
And there's a really important clinical example of this, right? Methanol poisoning.
Yes, a classic case. Methanol itself isn't super toxic, but alcohol dehydrogenase converts it to formaldehyde, which is very dangerous. We treat methanol poisoning by giving the patient large doses of ethanol drinking alcohol,
Why ethanol?
because ethanol is also a substrate for alcohol dehydrogenase, and it competes with methanol for the active site. By loading up on ethanol, you competitively inhibit the enzyme from processing the methanol, giving the body time to excrete the methanol harmlessly.
Brilliant. Okay, contrast that with non-competitive inhibition. How is that different?
In non-competitive inhibition, the inhibitor doesn't bind at the active site. It binds to a different site on the enzyme, often called an allosteric site.
Okay, somewhere else entirely.
Right. And binding there causes a conformational change in the enzyme that affects the active site indirectly, making it less efficient or completely inactive, even if the substrate can still bind.
So it messes up the catalysis part, not necessarily the binding part.
Often, yes. And the key kinetic effect here is that Vmax is lowered. The enzyme simply can't work as fast no matter how much substrate you add. Km is typically unchanged. The inhibitor doesn't affect the enzyme's affinity for the substrate, for those enzyme molecules not bound by inhibitor, just its ability to process it quickly. And critically, you cannot overcome this type of inhibition by adding more substrate.
Any common examples?
Heavy metal ions like mercury or lead Py2+ often act as non-competitive inhibitors. They tend to bind strongly to sulfhydryl SH groups on cysteine residues, which can be crucial for enzyme structure or function, disrupting activity.
Nasty stuff. And finally, the most permanent kind, irreversible inhibition.
Yeah, these inhibitors are different. They don't just bind temporarily. They form a strong, usually covalent bond with the enzyme, permanently inactivating it. The enzyme is essentially destroyed.
For good examples.
Unfortunately, some potent toxins work this way. Organophosphorus compounds found in some pesticides and nerve gases like DFP, diisopropyl 4-phosphate, irreversibly inhibit acetylcholinesterase, an enzyme crucial for nerve function.
And
Penicillin is actually a great example. It acts as an irreversible inhibitor, sometimes called a suicide inhibitor of bacterial transpeptidases, enzymes that bacteria need to build their cell walls. No cell wall synthesis, the bacteria die.
Okay, so enzymes are incredibly powerful, and they're involved in almost everything. It makes sense the body needs really tight controls to manage these metabolic pathways. What are some key ways enzyme activity is regulated?
Oh, absolutely. Regulation is critical. One of the most important mechanisms is allosteric regulation. Many key regulatory enzymes are allosteric enzymes. They have, in addition to their active site, one or more separate sites called allosteric sites.
Okay, allo meaning other.
Exactly. Modulator molecules, which can be activators or inhibitors, bind to these allosteric sites, not the active site. This binding causes a conformational change that either increases or decreases the enzyme's activity.
And these allosteric enzymes often behave a bit differently kinetically, right?
They do. Because they often consist of multiple subunits that cooperate, they don't typically show the standard hyperbolic Michaelis-Menten curve. Instead, they usually display a sigmoidal or S-shaped velocity curve. This allows for more sensitive regulation, Small changes in modulator concentration can cause big shifts in activity.
And a really important type of allosteric control, especially for efficiency in the cell, is feedback inhibition.
Precisely. This is a specialized, very common form of allosteric inhibition. Here, the final end product of a metabolic pathway loops back and binds to an allosteric site on the first enzyme unique to that pathway, usually the rate-limiting step.
So the product shuts down its own production line when enough has been made.
Exactly. It's incredibly efficient. A classic example is CTP, the end product of pyrimidine synthesis, inhibiting the enzyme aspartate transcarbamylase, ATCase, which catalyzes the first step. The cell avoids wasting energy and resources making something it already has plenty of.
Very neat. Okay, let's move right into clinical practice now. Diagnostic enzymes. This is where understanding enzymes becomes immediately practical for diagnosis. The idea is that enzymes leaking out of damaged cells into the blood can tell us where the damage is. Let's trace that classic pattern we see in the blood tests after a myocardial infarction, a heart attack.
Right, the sequential rise of specific enzymes in the plasma is absolutely critical for diagnosing and monitoring an MI. The first one we look for, the earliest reliable indicator, is creatine phosphokinase, CPK, but specifically the MB isoenzyme. We call it CPKMB or CKMB.
Why the MB isoenzyme?
Because CPK exists in different forms, or isoenzymes. The MM form is mostly in skeletal muscle, BB is mostly in the brain, but the MB form is highly concentrated in heart muscle. So a rise in CPK-MB is quite specific for cardiac damage. It typically starts rising within 6 to 18 hours after the heart attack.
Okay, CPK-MB first. What comes next?
Following CPK-MB, we usually see a rise in aspartate transaminase, AST, which you might also see called SGOT. This peaks a bit later, usually around 48 hours after the MI.
And the last one in the classic trio.
That would be lactate dehydrogenase, LDH, specifically the LDH1 isoenzyme. LDH also has multiple isoenzymes, 5 actually, made of different combinations of H heart and M muscle subunits. LDH1, which is HHHs, is the predominant form in heart muscle. It rises later than AST, maybe by day 2, but it stays elevated the longest, sometimes for 10 to 15 days, which can be useful for diagnosing an MI that occurred several days earlier.
That sequence, CPKMB, then AST, then LDH1, is really key to remember for timing.
Absolutely.
Okay, besides the heart attack pattern, there are other really important diagnostic enzyme markers. Can we quickly run through a few key ones and the organs they point to?
Sure. If you see elevated alanine transaminase, ALT, also called SGPT, that's highly suggestive of liver disease, think hepatitis or cirrhosis. ALT is more specific to the liver than AST.
Okay, liver, ALT, pancreas.
For acute pancreatitis, the key enzymes are amylase and lipase. Elevated levels of both are strong indicators. Elevated acid phosphatase, ACP, particularly the prostatic isoenzyme, was historically a marker for prostatic carcinoma, though PSA testing is more common now.
And one more, a sensitive indicator related to alcohol.
That would be gamma-glutamyl transpeptidase, GGT. While it can be elevated in liver disease too, it's particularly sensitive to alcohol consumption and can be a marker for alcoholism or alcohol-induced liver damage, as well as obstructive jaundice.
That's a fantastic overview of the diagnostic side. So taking a step back, the huge clinical significance here is that enzymes aren't just diagnostic markers, they're also major targets for drugs.
Oh, definitely. So many treatment strategies revolve around manipulating enzyme activity. We can target their synthesis or their activity directly.
Like cholesterol drug.
Exactly. The statin drugs, like lovastatin, are competitive inhibitors of HMG-CoA reductase. That's the rate-limiting enzyme in cholesterol synthesis. By inhibiting it, you lower the body's cholesterol production.
And for conditions like gout?
For gout, which involves excess uric acid, the drug allopurinol is used. It acts as a suicide inhibitor. The enzyme xanthine oxidase actually converts allopurinol into alloxanthine, which then irreversibly binds to and inhibits the enzyme, reducing uric acid production.
Clever. Even breaking down clots.
Yes, thrombolytic therapy uses enzymes. Drugs like streptokinase, originally from bacteria, and urokinase activate plasminogen to plasmin, an enzyme that breaks down fibrin, the protein mesh that forms blood clots. They're used therapeutically to dissolve dangerous clots in heart attacks or strokes.
It really highlights how central enzymes are. Okay, time for a quick knowledge check and then a final thought for you to ponder. First, the quiz question. Can you quickly recall which specific isoenzyme of creatine phosphokinase is that earliest, most reliable indicator we look for in the ER when suspecting a myocardial infarction?
That would be the CPKMB isoenzyme, also called CKMB. Its early rise is crucial.
Perfect. CPKMB. Got it. Now, here's a final thought to chew on. We've established that the enzyme's protein structure, the apoenzyme, is what makes it incredibly specific for its substrate. But coenzymes, like NAD plus stab, we said they're involved with hundreds of different enzymes carrying out the actual chemistry, like transferring electrons. So why isn't the coenzyme the thing that determines the enzyme's overall specificity?
Ah, that's a great question. It really comes back to the apoenzyme's active site. That intricate 3D structure of the protein forms a highly specific binding pocket. Think of it this way. The apoenzyme creates the unique lock, the active site, that only accepts a very specific key, the substrate. It's responsible for recognizing and binding the correct substrate and orienting it perfectly. The coenzyme might be the tool used in the reaction, like the screwdriver turning the screw, but the apoenzyme is the hand guiding the screwdriver to the exact right spot on the correct screw. Specificity comes from the protein part, the apoenzyme.
Excellent explanation. The protein dictates the who and where. The coenzyme handles the what, chemically.
You can put it that way, yeah.
Great job. You've just mastered another really key concept in biochemistry. Understanding these foundational workhorses, the enzymes, the kinetics, their regulation, and their diagnostic roles is truly essential as you build your medical knowledge. Keep up the great work.